Our first speaker is Pratap Tomar from the University of Bristol, and he's going to tell us, sorry, let me get your title, about altered hippocampal neural dynamics in a rodent model of chronic stress. Take it away.
Thank you. So can you see my screen?
Yeah, that looks good.
Okay. So
Great.
Hi everyone. I'm Pratap from University of Bristol. And today I'm going to talk about the impact of chronic stress, that is repeated exposure to stress, on hippocampal physiology. Hippocampus, as you may all know, is this medial temporal lobe structure shown in red here. It plays a crucial role in consolidation of information from short-term to long-term memory and spatial navigation. And in today's talk, I aim to show you some data that suggests that after chronic stress, hippocampal neural network, it undergoes it becomes a little synchronous, and that has implication for subsequent synaptic plasticity and learning and memory. This work was done in Riken Brain Science Institute in Tom McHugh's lab. Tom is here. And Dennis, this guy helped me with some of the analysis. And I also want to acknowledge Professor Shona Chatterjee from NCBS India, who helped me, who generally is expert in stress field. So we keep talking to him about our analysis and results. And currently I'm working in University of Bristol with these two wonderful professors, Jack Miller and Matt Jones. Okay, so let's get started. It's well established that that the stress affects cognitive processes, including learning and memory. And these animal stress models have managed to capture some aspects of it. So for example, using chronic restraint stress, it's been shown that hippocampus dependent learning gets affected. For example, in Morris water maze tasks, these stress rodents, they take much more time to escape this maze. So this is work by these Park et al. From this is not my work. So they consistently take more time to escape the maze. So latency is higher, right? So stress affects hippocampus dependent learning and memory. And if you take brain slices from these rodents, then LTP or synaptic plasticity is also impaired, such that their capacity to undergo synaptic plasticity decreases. Yeah, and in late 1990s and early 2000s, people, when they looked into hippocampal neural structure, they found that pyramidal neurons, they undergo dendritic shrinkage. So dendrites are shorter and there are fewer spines on them. And in today's talk, this is my work is around, so in vivo, I'm going to show you some in vivo recordings where I'll show you that neural activity as well as network activity, which means local field potentials, how they get affected after repeated stress. So a little bit about in vivo electrophysiology of hippocampus. So hippocampal physiology looks different based on what behavior is going on. So for example, if we record LFPs from hippocampus, during online states, that is when brain is engaged with surroundings, for example, during exploration, you see this very nice rhythmic theta activity. So this rhythmic activity, and this is roughly 6 to 12 hertz. However, during offline states, when brain is not engaged with surroundings, that is during rest and non-REM sleep, that's when you see these very high frequency, so this large irregular activity. And if you zoom into one of these, then you see these high frequency oscillations, oscillatory transients called sharp wave ripples. And previously I have looked at this during these theta oscillations, hippocampal neural activity, and found that chronically stressed subjects, their ability to encode new environments get affected, such that in chronically stressed subjects, their place maps or internal representations of these tracks, they look very similar. So, and in today's talk, we are going to focus on these sharp wave ripples. And previously it has been shown that these ripples are important for consolidation of information. So for example, if you detect the start of the ripple and you inject little currents such that they don't run their course, then these subjects, they perform poorly on a memory task. So there are enough studies that have shown that. So ripples are very important for information consolidation. And in today's talk, So I'm going to show you is we are using a chronic immobilization stress. That is, animal undergoes 2 hours of immobilization stress for 10 consecutive days. And I'm going to show you recordings during stress state. So how different that neural activity is in comparison to offline state on day 1, and then same on day 10 on the last day of stress. And what I'm interested in is how neural activity is different between day 1 and day 10 during rest state, right? So this is where chronic stress would have done its job. And then, so question is, after repeated stress, how different the neural activities. And all my recordings are from area CA1 of the hippocampus, which is the output region of the hippocampus. So it makes sense to record from this. So let's dive into the results. First, so here I'm showing you a cluster of cells during rest state and same cells during stress. So each cluster, each color represents one cell, and each cluster or each color is made up of many, many dots. So each dot is one action potential. So you can see there are 8 to 9 cells, and so this cell is highly this fires more, maybe this cell fires less. And during stress, same cells, their firing rate decreases. So their activity is suppressed during stress. Here is the quantification. So y-axis is mean firing and x-axis is time, so day. So this is day 1 and this is day 10. Black is rest, red is stress. So during stress, you have a robust decrease in spiking activity of these pyramidal cells. And on day 10, even during rest state, you find a small decrease in firing rate. And then we don't see a further decrease or change. So then we looked into ripple activity. So during ripples, what happens to these the pyramidal cell activity? And so now we are looking at pyramidal cell activity specifically during these sharp wave ripples, and we find exactly same phenotype. So during stress, the firing rate is less ripple associated firing rate. And then on day 10, during rest, you have the same phenotype. And then there is no further decrease. Then next we looked into the participation of these cells into ripples. So usually cells, they spike. They can fire inside the ripple and they can fire outside. And as an average, they fire in certain a percentage or fraction of ripples. And what we find is that similar to similar to the pyramidal cell activity, their participation decreases. That is, during stress, they participate in fewer ripples. And on day 10 itself, then there is a decrease in the participation. So whatever happens during stress, you can start seeing same phenotypes occurring even during rest state on day 10. So now then we looked into the coactivity of these cells. So if cells are firing less in ripples and they are participating in less, then is their coactivity also decreasing? And to our surprise, we found an increase in coactivity. So during stress, the coactivity is enhanced, and that is also true for day 10 during stress, right? However, importantly, from day 1 to day 10, you can see that there is an increase there. So that is what is very interesting. So what is happening is somehow pyramidal cells, they are becoming, though they are firing less during ripples, but their coactivity is higher. So they are becoming, the network is becoming more coordinated during stress. We also looked into other properties such as ripple length and amplitude, and we find that during stress, ripple length increases. So ripple duration is high, both on day 1 and day 10, and there is no difference between rest state from day 1 to day 10. So this is ripple. So this is stress specific phenotype. This is what happens. We also looked into other ripple properties like ripple occurrence. So ripple interval, that did not change, and same was true for frequency within ripple. So peak frequency, that did not change. So what we think is occurring is that when animal is resting, then you have these different cells. They are active in the ripple, they are active outside ripple, and they become coactive in certain percentage of ripples. So they become coactive. And after repeated stress, we think what is happening is generally their firing rate is decreasing and their participation is also decreasing during these ripples. However, as an average, their coactivity is increasing. So hippocampal networks becomes much more coordinated or synchronized during these ripple states. And ripple length itself is higher during first exposure to stress. So this is what I have already sort of mentioned. And what happens after 10 days of 9 days of repeated stress on day 10 itself, that some of these stress phenotypes you start seeing even during rest state when no overt stress is delivered, right? So, and then what is happening is that if you compare it from day 1 rest to day 10 rest, the hippocampal network activity is much more coordinated or synchronized. So in conclusion, what we found is that repeated stress led to increased synchrony in hippocampal neuronal network. And idea is that, so now how we think about it is that if neurons are becoming active at the same time, then this will lead to some kind of synaptic saturation if it is occurring, continuously. So because cells that fire together, wire together, there will be sort of, so maybe because of that synaptic saturation, there will be a decrease in if synaptic saturation happens, then their capacity to undergo subsequent synaptic plasticity or LTP will be impaired. And that may have consequences on learning subsequent information. And maybe that is why, we find that their ability to differentiate, make representations of these two different surroundings gets affected. So place cell remapping gets affected. With that, I thank you all for listening. And this work is currently in bioRxiv. Hopefully, if reviewers are kind enough, to get published soon. So yeah, so that's pretty much it. And I'll be happy to take if you have any questions.
Great, thank you. That was really interesting. And thank you for keeping on time. We have a question from...
So should I stop sharing?
You can keep it on since you might need to refer to slides.
Sure. Yeah.
We have a question from Alina Jacobs. Do you have any non-stress controls? I believe in chronic recordings, the quality of recordings may decrease over time.
Well, in this case, we have not done that, but the question would be that firing rate will decrease. Is that the question?
Yeah. So just how can you rule out the fact that just across the 10 days in your chronic preparation, you might see sort of the clusters become, yeah, exactly. The clusters become poorer and your firing rate looks to be lower.
I see. So that information is there in the manuscript. So if we look at, for example, these cluster quality, so we do cluster quality, for example, these L ratio and isolation distance, that itself does not change. So cluster quality is not changing, that we are very confident about. So now, question is somehow firing rate changes. I am not sure how that will happen, because what happens is that, our tetrodes sort of, is not stationary, right? We just sort of keep moving them up and down. So it's not that that it's at one place and we are not able to record neural activity. So I really doubt that that is going to affect firing rate.
Okay, great. I have a question.
Yeah.
How permanent is this effect? So you have this 10-day stress exposure. For how long thereafter do you see what appears to be permanent changes to the network?
So that is a really good question, and I have not looked into that long-term effect yet. What is known, for example, in terms of this dendritic atrophy that I talked about in hippocampal pyramidal neurons, that stays for some time, and then after 21 days, if you give it a 21 days recovery, then dendritic shrinkage goes back, in the sense that the dendrites are normal. So honestly speaking, I have not done that experiment, so I won't be able to answer that with confidence, but it should last for some time. So I have not done that experiment.
That's fair enough. Can you speculate?
Well, the thing is that if it is a long-term change. So for example, if it's happening because of your hormonal levels going up, for example, corticosterone levels and even the norepinephrine levels, till they will be up, I think you will see this enhanced synchrony, which should last at least, for some time. And I'm sure after, like dendritic atrophy, after a couple of weeks, this should come back. So I think this effect, this enhanced synchrony will be around till you have that chronic stress lasting and maybe few days, not after a very long period of time. So this should not be permanent.
Fair enough. Okay, I have more questions, but we're out of time. So thank you, Pratap, and we'll give over the screen share to our next speaker, Tim.